So thank you for your interest in scientific research being done at the Berkeley Lab, especially those of you that haven't been here before. We want to thank our many co-sponsors, UC Berkeley, the Chabot Space and Science Center, the Exploratorium, the Osher Lifelong Learning Institute at Berkeley, the Lawrence Hall of Science, and the science departments of Albany, Berkeley, and Oakland High Schools. I'm very pleased to introduce Dr. Carolyn Bertozzi, Director of the Berkeley Lab Molecular Foundry, and the T.Z. and Irmgard Chu Distinguished Professor of Chemistry and Professor of Molecular and Cell Biology at UC Berkeley. Dr. Bertozzi completed her undergraduate degree in chemistry from Harvard and her PhD from Cal in 1993. After her postdoc work at UCSF, I found out she was the Department of Anatomy, where I used to work actually, in the field of cellular immunology. She joined the faculty at Cal. Professor Bertozzi has been recognized with many honors and awards for both her research and teaching accomplishments. She's a member of the National Academy of Sciences, the American Academy of Arts and Science, and the German Academy of Sciences. She was a MacArthur Fellow and received research-related funding from a number of organizations. And actually, we're pleased tonight to have Jan Katz from the American Cancer Society here. I don't know, Jan, if you can raise your hand. She's got a table out in the lobby if you want to visit. They actually funded your postdoc work, is that right? Oh. When you were at UCSF. And as Ms. Katz tells us, 42 of the folks that they've funded in the past have actually won Nobel Prizes, so you're in good company. And maybe that's a foreshadow. Professor Bertozzi participates in high school outreach programs. I know we have some students from Berkeley High here tonight. Are you? Raise your hand, I can see you there, yeah? And as well as programs that promote the participation of women in science. And tonight she'll be reaching out to all of you with her talk about body sugars and what they can tell us. So welcome, Dr. Bertozzi.
Thanks. Thank you. All right. I was thinking we could make a deal. I'll take a shot at the Nobel Prize if you guys are willing to go on YouTube with your questions. I'm not sure which is harder. Okay. So first of all, let me extend my gratitude to all the organizers of this event. It's just fantastic. You know, I've seen the advertisements around town now for at least a year, and it was a great privilege to be invited to come up here and have the stage and have your attention for the next hour. And to tell you about some work that's been going on in my laboratory for over 10 years now. We've been chipping away at a difficult problem that has to do with cancer diagnosis. And I'll show you how far we've come. We certainly have a long way to go. And so it's a great privilege for me to stand up here and represent my laboratory. And of course, all the hard work is performed by my graduate students and my postdoctoral fellows and my undergraduates at UC Berkeley and at the Lawrence Berkeley Lab. So I can't remember the title that's on the actual poster, but we're going to talk about sugars tonight. And everybody here has certainly heard the term sugar. I mean, when you hear the word sugar, you think of that white packet of stuff from Starbucks that you put in your coffee, right? Or you sprinkle it on your cereal, or maybe you cook with it. You think of sugar as something you eat, and generally you probably think of sugar as something that's bad for you. Hence, you know, such fads as the low-carb diet, and all of us are trying to cut back on sugar, and there's all these artificial substitutes, and so on and so forth. So what I'm going to talk about tonight, and in fact what we work on in my laboratory, is not so much sugars as a food substance that's bad for you, but rather how we can use sugars in a way that could be very beneficial for the diagnosis and potentially the treatment of human disease. So it's kind of a different kind of sugar that we're going to talk about tonight. But let me just introduce the topic a little more generally, starting with the way that I first learned about sugars in biology. Okay? And it takes me back to a lecture that I heard when I was in college. And so I took a cell biology course back in 1980. And my professor at Harvard at the time said, well, here's what you need to know about sugars. Sugars are coating the surface of your cells, and so he drew up on the blackboard a cartoon of a peanut M&M, and he said, this is what you need to know about sugars, that's all you need to know, the end. Okay? They're like a shell surrounding your cell. And in fact, if you bit through the middle of that peanut M&M, you'd see something like this in cross-section. There's a sugar coating, then there's that layer of chocolate. And with respect to the cell, we think of that as the lipid bilayer, this kind of a layer of fat on your cells. And then inside the cell, there's all kinds of proteins and interesting complicated things. And that's what we learned at Harvard University, okay? So I thought that was pretty interesting, and I remember asking my professor in class, well, why are cells coated with sugars? To which my professor responded, well, it's just like the peanut M&M. It's there because, like with an M&M, the sugar coating makes sure that the M&M melts in your mouth, but not. Yeah, everyone remembers that cartoon. So that advertisement was in its prime, back in the 1980s. And so basically we were taught that cells are coated with sugars because it protects them. It's like a physical barrier so that cells don't disintegrate or melt or something like that. So that was then, 20 some odd years ago, and the field has come a long way since then. And nowadays, if you look at the modern textbooks for cell biology, you'll see cartoons that look more like this. So this is an actual electron micrograph of a cell. So it's like a section through the middle of a living human blood cell. In fact, this particular blood cell came out of the body of one of my graduate students. He took his blood, pricked his finger, and went and took this beautiful image so that we could look at his cell in all of its glory. Now, the cell is bounded by this structure called the plasma membrane, which is like the layer of fats. It's like the chocolate on the M&M. And that's what this cartoon represents. But then it turns out that embedded within this lipid bilayer are a variety of molecules that we call glycoproteins and glycolipids. And this Greek prefix glyco means sugar. So the bottom line is that there are proteins and lipids and all kinds of molecules that are embedded in this layer of fat. And they're basically projecting a forest of sugars on the surface of your cell. So if this cartoon looks a bit like trees planted in the earth, that's no accident. That's probably what your cells look like. They're like a planet, and there are forests, and some of those trees are very tall, and other trees are sort of short, little shrubs. But the forest is made up of sugars. Now, the cartoons I'm showing here are what we in the field would refer to as complex sugars, which you have probably heard referred to as complex carbohydrates. These are the same thing. Carbohydrates are sugars, it's interchangeable terms. The complex sugars are made up of simple sugar building blocks. And these are like the smallest units of sugars. We call them monosaccharides. That's the technical term. But they're building blocks, and I thought I would show their chemical structures for anyone in the audience who's sort of interested in that kind of thing. But we give them each a name, and sometimes you can even see these names in the ingredients list of your cereal box. Okay, these are simple sugars. They're found in many of the foods that we eat. And in your body, these simple sugars get linked together in chains to make the complex sugars, or the complex carbohydrates. So I thought I would just introduce some terminology. And again, these are words that you may have heard before, but you might not realize that it has to do with sugars. So each of these simple building blocks we call a monosaccharide. And if you link together two monosaccharides, you get a disaccharide. And probably the most famous of all disaccharides is the milk sugar that's known as lactose. So lactose is what you get when you take this green monosaccharide and you link it in a particular way to this blue monosaccharide. Now, lactose, of course, is a wonderful substance for those of you who can tolerate it, but many of us can't. So how many people are lactose intolerant? So am I. And what that means is that we are all missing an enzyme that's able to split the disaccharide into the two monosaccharide building blocks. Now, the monosaccharides are readily taken up in our intestines, and we use them, as nutrients for energy. But the disaccharide cannot get absorbed through our intestines. So if you don't have the enzyme, you can't split the disaccharide. You can't absorb the disaccharide, and the result of that is you end up with pressure in your intestines, and fluid rushes in there, and it's just a disaster, and we don't have to go through the details. But in any event, you get the picture, okay? That enzyme is called lactase, and the good news is that you can buy it off the shelf and just kind of, dribble it into your latte or just take a pill before your pizza, and usually you're okay. Now, if you take many, many, many monosaccharides and link them together to make very, very large oligo or polysaccharides, then you can get some very interesting materials, some of which are like the building blocks of nature. I thought I would point out cellulose as an example. It's a very long polysaccharide that's made up of very simple monosaccharide building blocks, and it's the most abundant organic material on the planet Earth. That's a good thing to know. Why is everyone so interested in cellulose? Well, it's a potential source of energy. And lots of folks, including Steve Chu, are interested in how we can convert cellulose to the monosaccharide unit, which is called glucose, which ultimately can be used to generate ethanol, which is a biofuel. So there's lots of interest in the chemistry of cellulose, and you'll certainly hear more about this, I'm sure, in other presentations at the Berkeley Rep, because there's lots of people at Cal that are working on cellulose. Okay. Now, if you look back through the history of this field of sugar biology, I would argue that the most important discovery that brought sugars to the forefront in the eyes of doctors and biologists and chemists and people who work in the clinic was probably the discovery that sugars are what make up what we now know as the human blood groups. So you know your blood type, right? It turns out your blood type is determined by the sugars on your blood cell, and it can vary between people. So how many people here are blood type O? Okay, it's quite a few people, so am I. What we all share in common, us blood type O's, is that we have this arrangement of sugars on our blood cells. We call this a trisaccharide because there are 3 of these monosaccharide building blocks and they're linked in a particular way. So that structure is defined as the blood group O. And we have that decorating the surface of our cells. Now, who's blood type A? Okay. Here's what you have that we don't have. You have an enzyme that can take this red sugar and put it on this green sugar to convert the trisaccharide to a now tetrasaccharide. That is defined as blood group A. Now, who's blood group B? Usually this is the most rare in the audience, so there's a few of you out there. You have a slightly different enzyme. Your enzyme takes another green sugar, puts it on the green sugar to make this tetrasaccharide. That's blood group B. Okay? Now, I don't know if you can tell by looking closely, and I realize the green, I think, is a little faint from your perspective, but if you look closely, atom for atom, at the A and the B structures, what you'll find is that it's a very, very small difference. If you look right here, there's an oxygen and a hydrogen in the blood group B, and in that same position, which is right here, in blood group A, there's different atoms. That's the only difference in these entire structures. Now, that seems almost insignificant from the perspective of a chemist, but the human immune system sees these two as night and day. Because if you take blood from a blood group B donor and put it into a blood group A recipient, it's a catastrophe. That blood group A recipient's immune system will see that blood as foreign, will attack the blood and destroy the blood, and you'll end up with hemolytic anemia, and probably it's a fatal condition. So, by understanding how the sugars govern the blood types, this enabled physicians to develop what is now the most important medical procedure in the world, which is the blood transfusion. You couldn't do a blood transfusion if you didn't understand the blood types. And that was a moment in the history of sugar biology when immunologists, physicians, chemists all came together to understand how, at the level of these molecules, are blood cells recognized as self or as foreign. It was a really important moment. It occurred in the late 60s, early 70s. That was a big deal in our field. By the way, somebody always asks me, what about AB? Who's AB? Okay, here's your story. You got this enzyme from one of your parents and this enzyme from the other one of your parents. You have both enzymes. You have a 50-50 mixture of A and B on the surface of your cells. That's the deal. Okay, it's all genetics. I noticed, by the way, that some of you did not raise your hand for any of the above, which means you don't know your blood type. You should figure that out. Ask your doctor. Okay. So we now know that not only do the sugars that coat the surface of your cell encode information about yourself, but they also relay information to the outside world. For example, there are now many different examples in human biology in which we know that the sugars, which are now these little red structures in this cartoon, that the sugars on one cell can interact with proteins on another cell. And the proteins literally grab onto the sugars, and this allows two cells to cling together. That's really important. Otherwise, your body would just fall apart into a bunch of dissociated cells, but that doesn't happen. You're walking around, you look good. That's because your cells are in contact with each other. They're clinging to each other, and they're using those sugars to do it. Now, that's the good news. Here's the bad news. The bad news is that bacteria and viruses have noticed that your cells are covered with sugars. They would like to cling to your cells too. And so many viruses and bacteria have their own proteins that grab onto your sugars, and they hang on, they colonize your tissues this way, and this can make you very sick. I'm going to show you an example in a minute. And then we also know that certain toxins can grab onto the sugars, and that's a bad thing. But then in your body, certain hormones can grab onto the sugars, and that can be a good thing. Okay, so the sugars are used every day by your normal biological system and sometimes exploited to your detriment by microbes in the world we live in. Take, for example, the influenza virus. It's a timely subject. This is flu season. How many people had their flu shot? Oh, this is really good. More of you had your flu shot than know your blood type, I've noticed. And hopefully this never happens to you. But what you're looking at here is a human cell that's been infected with the influenza virus. The virus has taken over the machinery of the cell. It's replicating itself and producing new daughter virus particles. So here's the human cell. It's sort of this big gray structure. And each one of these little balls is a new nascent viral particle that's about to bud out of the surface of this poor hapless cell. And here's a free particle that probably just left the nest. Now, we know quite a bit about how the influenza virus works. And it turns out the life cycle of this virus is all about sugars. So let me show you. Now, here's a cartoon. So each one of those virus particles can be depicted something like this, where it's got a plasma membrane coating, it's got a lipid bilayer, and then it's got a whole bunch of proteins and nucleic acids in the middle, which I'm depicting with this sort of funny red shape. But there's two important proteins for the viral life cycle that basically govern how it gets into your cells and then how it gets out of your cells. And those two proteins go by the name of hemagglutinin and neuraminidase. Hemagglutinin is what we call a receptor. That means it's kind of like a claw, and it's able to grab a sugar on the surface of your cell that goes by the name sialic acid, and I'm abbreviating that SA. So your cells have this sialic acid. The virus has this claw, and the claw can grab that sugar. That's how it's going to attach itself to your cell, which helps it get into your cell. This other protein, neuraminidase, is what we call an enzyme. This enzyme is like a pair of scissors. The enzyme is going to cut the sugar off of the surface of the cell. Now, when scientists discovered the hemagglutinin and the neuraminidase, it was like a big question mark, because it seems paradoxical. Why would the virus have one protein that's like a claw that hangs onto a sugar, but another protein that's just a pair of scissors that cuts the sugar off? Well, it started to make sense when the life cycle of this virus was basically broken down to its various stages. So at the very beginning, here's the virus. It comes down onto the target cell basically like a lunar module landing on the moon. And this target cell is a cell in your nasal cavity or in your upper respiratory system because someone just sneezed next to you on the BART and you happen to breathe it in, and there you are getting infected. Now, when the virus lands, that claw grabs the sialic acid, and that's the very first step in the infection. Once that happens, it allows the virus to enter the cell through a process that's called endocytosis, which is basically like the cell eating the virus. Now, once the virus has made it into the cell, it can set itself loose and start to replicate. So many, many, many little daughter viral particles are born. Those particles then snuggle up to the surface of the cell where additional copies of hemagglutinin and neuraminidase are biosynthesized, and then a whole new viral particle is formed and it sort of buds off of the surface. But now, that hemagglutinin, that same claw that allowed it to attach in the first place, is still holding on. And that prevents the virus from letting go so it can find the next cell and infect the next person, and so on. That's where neuraminidase comes in. That pair of scissors is there to cut the sugar off so the virus can set itself free and travel to the next cell in the infection process. So both of these proteins, the hemagglutinin and the neuraminidase, are absolutely critical for the ability of this virus to proliferate. And for this reason, a number of biotechnology and pharmaceutical companies have been interested in making drugs that block these two proteins. And where they have had the most success is in the neuraminidase area. So let me just show you some drugs that are actually on the market today. And it turns out that they're based very much on the structure of that sugar, sialic acid. So remember, neuraminidase is able to recognize that sugar, and like a pair of scissors, it sort of cuts it loose from the surface of your cell. Well, the two drugs that were made by two different companies bear quite a bit of resemblance to that sugar molecule. The first of these is called Influenza, and I think that one was developed by GlaxoSmithKline. And you can see some of these parts of the molecule look very similar, very similar. There's a little bit of difference here that's important for the way that the drug behaves. But Influenza is a pretty decent anti-influenza drug. But it has to be administered through inhalation, so you get a little inhaler. And then along came this other drug named Tamiflu. This one was developed by Gilead Pharmaceuticals, which is right here in South San Francisco. And it also, you can kind of see the resemblance to the sugar. There's bits and pieces that look similar. Tamiflu is a little bit better than Influenza because you can take it as a pill, which people find a little more comfortable than inhaling the drug. But don't give up your flu shot just yet. And I'll tell you why. Because neither of these drugs is very effective for somebody who already has the flu. In fact, most of the time these drugs are used for patients before they get the flu, to prevent the flu. And you might think, well, who has got the ESP required, to determine if you're going to get the flu next week? Well, the way that this works is if your spouse has the flu, maybe you should go on one of these drugs, to prevent yourself from getting it, maybe your kids from getting it. If there's an epidemic in a nursing home, maybe all the people who haven't got the flu yet, maybe they should go on these drugs. So that's how these drugs are used. But you've got to take them before you actually get sick, otherwise they don't work too well. In other words, get your flu shot. Okay. So this is one example in which understanding the biology of the sugars with respect to influenza has allowed us to develop drugs to actually treat the disease to some extent. So here's an example where sugars can be good for you. But what we study in my lab is something actually very different. We're interested in cancer. And it turns out over the last 20 years or so, a number of researchers have discovered that the sugars on cancer cells can be very different from the sugars on our normal, healthy cells. That somehow, when cells start to transform from a healthy state to a malignant state, the transformation is reflected in their cell surface sugars. We don't really know why that is. It's a very interesting observation. But it is the fact that if you take a tissue from a human being who's got cancer, this is a liver. This is from Google Images, okay? This isn't from my laboratory. But if you take some of the normal healthy tissue from this tumor-infiltrated liver and just look at the pattern of sugars and compare that to the cells that are coming from the cancerous regions of that very same tissue, you'll see that the patterns of the sugars have changed. It's as if the forest has been remodeled. New trees have grown. Old trees have died. Okay? It's a completely different landscape. And this has been known now for almost 20 years, and the evidence keeps building. So we have been intrigued by this observation, and it begs the question, if cancer cells' sugars look different from normal cell sugars, maybe we can exploit those differences to detect cancer inside the body. But that would require that we can somehow see those sugars. I mean, literally visualize those sugars in the human body. And that has been the challenge that the students and postdocs in my lab have taken on over the last 10 years or so. Now, one of the changes in sugars that occurs on cancer cells versus normal cells actually has to do with that same sugar that the influenza virus is recognizing, sialic acid. Okay, it's kind of a coincidence. In fact, if you look at some of the complex sugars that are known to appear on cancer cells, and I'm showing some of their chemical structures here, you'll note that, at least in this slide, and many I haven't shown, that these structures have that same sugar, sialic acid, as one of their components. And I've highlighted that in green. Other aspects of these structures are actually quite different, but they do all tend to share this common feature. They have that sugar sialic acid. And what this means, if you just kind of inventory all the sugars collectively, is that cancer cells tend to have a lot more sialic acid than their normal, healthy counterparts. And the differences sometimes can be pretty low, maybe 10 times. We don't consider that particularly dramatic. But in other circumstances, the differences can be quite spectacular. You can have a 1,000-fold increase in sialic acid on certain leukemias compared to normal healthy blood cells. You can have 1,000 times more sialic acid on certain brain tumor cells compared to normal brain tissue. So if there was some way that we could see sialic acids in your body, maybe we could detect the presence of these tumors. And hopefully, when they're still at a very small stage, when they're much more easily treated than larger tumors. So we imagined a scenario in which we might be able to actually see these sugars using the standard clinical imaging techniques that many of us are already familiar with. For example, many of us have had a magnetic resonance image taken of some part of our body. This is now considered a fairly routine clinical imaging technique, an MRI. How many people have had an MRI? So, see? You see what I'm saying? Probably fewer of us have had a PET scan, but it's also quite a standard clinical imaging technique. And then even fewer of us have probably had a fluorescence image taken of our body, but nonetheless, fluorescence imaging is something that's sort of becoming more common practice in the clinic. And the bottom line is that we have these wonderful techniques available to us that allow us to see inside the human body without cutting ourselves open. And these techniques have been very powerful mechanisms for trying to find tumors, but they're not particularly sensitive, and they pay no attention to sugars. So if we could somehow direct these imaging techniques towards the sugars, maybe this would give us a new window that we could look through in order to find these cancers as they're forming. Now, there's no way to see the sugars as they are in their normal state. They're basically invisible to any of these imaging techniques. And so we thought maybe we can use chemistry to make the sugars visible, which means that we have to somehow direct what we call an imaging probe to the sugars so that we can see them. And hopefully that will become evident in a minute. And in thinking about how could we put imaging probes on these sugars, the next question was, how are the sugars made in the first place, and can we actually exploit the natural biosynthesis mechanisms in the body in order to achieve this goal of seeing sugars? So I thought I would just take a minute to remind everybody about where these sugars come from. I think this is probably self-evident, but you are what you eat. So most of the sugars in your body are coming from your diet. So whatever you had for lunch and for dessert had some sugars in it. Those sugars are taken up by your cells after they enter your body, and then your cells transform them through a variety of enzymatic processes to generate those monosaccharide building blocks. Now, the building blocks end up inside a part of the cell that's called the endoplasmic reticulum and the Golgi compartment. And the details here are not important, but it suffices to say that this part of the cell is an assembly line, like at General Motors, okay, where one by one, the monosaccharides are linked together to make these complex carbohydrates. And then finally, all of that business is delivered to the surface of the cell, and those sugar trees are projecting out to the cell surface. But to summarize all of this, you eat these little building blocks. Those little building blocks end up in these trees. So we thought maybe we could put something in the building blocks that allows them to be visualized when they're exposed on the cell surface in the form of these trees. So we came up with this very simple idea. And the idea was to take a synthetic sugar that we had modified with a chemical group. We're going to call that chemical group X for now. That's just a generic X. We're going to feed cells, or human patients, that synthetic sugar. And if the synthetic sugar looks enough like your natural sugars, then the enzymes will simply transform that sugar inside the cell, and it will become incorporated into those trees on the cell surface. And that chemical group X will hopefully be ignored altogether by your cells, just come along for the ride, and end up displayed in the trees on the surface of your cell. Now, that's step one. Step two is where we introduce what we call an imaging probe. The imaging probe is a molecule that can be visualized by MRI, or by PET imaging, or by fluorescence imaging. And that probe molecule will have its own chemical group, Y. And Y, again, I use this generically, Y will be chosen so it can form a bond, do a chemical reaction with X, so X and Y are now linked together, and that positions the probe on the sugar that we're trying to visualize. So overall, it's a two-step process. The first step we call feeding. We just feed the body an unnatural synthetic sugar. The second step is the chemistry. We react that sugar with an imaging probe. So now we can see it on the surface of the cells inside your body without cutting you open. That's the idea. Okay. So in concept, I think this is rather simple. In practice, it's taken us like 12 or 13 years, okay, to try and make progress here. And the fundamental challenge has been in the choice of X and Y. Because X and Y are two chemical groups that have to come together and form a bond. Now, that might not seem like a big deal, but remember that they have to form that bond inside your body. And that's the hard part. Because there's so much stuff in your body. There's so many molecules with so many different kinds of chemistry that for X and Y to ignore all of that noise and only find each other across a crowded room and form a bond, ignoring, parting the sea, this is the hard part. And this is what has taken us so long to work out. But I think we've got the chemistry. And it was inspired, ironically enough, by a chemical reaction that was published over 40 years ago by Rolf Huisgen, who was a German chemist. He was at the University of Munich. Now he's retired, but he's still around, still on the circuit. And back in the 1960s, Rolf Huisgen discovered that this chemical, which is known as an azide, it's 3 nitrogen atoms bound together in a line, that group, that functional group, as we call it, will react with this chemical group, which we call an alkyne. An alkyne is 2 carbons that have a triple bond. When N3, the azide, and the alkyne get together, as long as you heat it up, you can get them to come together into a single product that's called a triazole. It's this 5-membered ring. These are the 3 nitrogens that came from these 3. These 2 carbons came from these 2. And then you've got this blue thing and these red things, and they're all kind of linked together now around this 5-membered ring. That's the triazole. This kind of chemistry seemed perfect for our needs because neither the azide nor the alkyne exist in your body. And furthermore, neither the azide nor the alkyne would react with anything that does exist in your body. So these are 2 functional groups that, in theory, could be injected into your body, ignore everything else, find each other, and boom, form a triazole. But there's one problem. It's this thing right here, okay? That's 100 degrees Celsius. That's boiling water, okay? And that's fine for Rolf Huisgen's chemistry in a test tube, but you cannot do that to a human patient, okay? So we knew that this was a showstopper. But it got us thinking about ways that we might be able to slightly alter this Huisgen reaction so that it would proceed not at 100 degrees, but rather at 37 degrees, body temperature or room temperature. And here's how we did it. It turned out to be a geometry problem. Okay? So it's not chemistry anymore, now it's geometry. So here's the deal. The reason this reaction requires so much heat is that if you look at the angle of the bonds here, it's 180 degrees. So these three things are in a straight line, okay, 180 degrees. If you look at this triazole product, okay, those same three things, which are now down here, there's an angle of 120 degrees. So you've got to take this molecule that's linear and bend it to get to 120 degrees. And that's hard to do. That's why you have to heat it up. You have to put energy into the system to get it to bend. So we thought, well, what if we change the structure of this alkyne so that it's already bent? And here's what we did. Okay? So this was the idea of one of my graduate students, Jeremy Baskin. And he said, well, what if we take this carbon-carbon triple bond and force it into a bent structure by sticking it in a ring? In fact, an 8-membered ring, he realized, is something that you can actually make, and it bends the angle to about 160 degrees here instead of 180. And that's closer, though not identical, but closer to where it's going to end up, which is at 120 degrees. And then he also discovered that if you put 2 fluorine atoms right here, that makes it even more reactive, and there's some physical reasons for why that is. But the bottom line is that by trapping this alkyne in an 8-membered ring, and then putting a couple of fluorine atoms, he was able to make a substance that now reacts with an azide to form that same kind of product at room temperature. And we call this general kind of chemical reagent, this difluorinated cyclooctane, that big mouthful, we just abbreviate that DIFO. Okay, so this is DIFO, invented by our own Jeremy Baskin, soon to appear in chemical catalogs near you. Okay? So the beauty of DIFO is that it takes advantage of what we call ring strain so that it reacts with this azide much more quickly. And now these two chemicals will find each other across a crowded human body, we think, and react without boiling that person in water. In fact, we've already done some experiments that suggest that these two reagents will in fact allow us to visualize sugars in living subjects. So for example, we started by doing some experiments, not in humans, but on human cancer cells, just in a culture dish. And so what we did is we took those cancer cells and we fed them this synthetic sugar, which is a metabolic precursor of sialic acid. It's one of the intermediates in metabolism in the cell. And we simply decorated it with the azide. Those are the 3 nitrogens. And when you feed this to cells, the cells very happily take it up, they process it, and lo and behold, on the surface of the cell starts to emerge sialic acid sugars with azides. So putting an azide into sialic acid is as simple as feeding the cells this synthetic sugar. And then the question is, if we take these cells that have the azides embedded in their sialic acids, can we actually see the sialic acid by doing a chemical reaction with that DIFO reagent linked to some kind of imaging probe? And if this reagent will find that azide and form that bond, then the probe should be on the cell and we should be able to see it. So the kinds of probes that we've been working with in our laboratory are fluorescent probes. And I'll show you, this is a molecule that was designed and built by one of my graduate students whose name is Pam Chang. What Pam did is she took that DIFO molecule and just linked it to this other molecule shown in green, which is a fluorescent molecule. What that means is that when you shine light on the molecule takes in the light and then shoots light right back at you of a different wavelength. In this case, it's a green color. So wherever we see green light, that's how we know that this molecule is localized on the cell. And what you're looking at here are fluorescence images of actual human cancer cells that are growing in a tissue culture dish, a little plastic dish. And in this top panel, we fed the cells that synthetic sugar with the azide. So these cells have sialic acid azides all over the surface of the cell, and that's why the cells are all outlined in green. Can you see that? Those are the sialic acids. And these blue circles, those are just the nuclei of the cells, just to prove to you there are cells there. Okay? We have a special dye for the nucleus. What's really important is this bottom panel. It looks a lot more boring. In this panel, instead of feeding the cells that sugar with the azide, we fed the cells the natural sugar, which has no azides. So these cells have regular sialic acid, no azides at all. And when you treat these cells with this molecule, there's nothing for it to react with. So that's why there's no green at all, just the blue nuclei can be seen. And that's very important because we don't want this molecule to attach itself to anything that doesn't have the azide, because the azide signifies the sialic acid. And that's all we want to see. We want to see sialic acids. So this was very encouraging. And after Pam was able to demonstrate that she could see the sialic acids on cells in a test tube, the next obvious question is, can we see the sialic acids on cells in a living organism? Now, of course, the way scientific research works is you cannot take molecules that you've tested on cells and immediately start shooting them into your undergraduate students. Okay? You can't do that. Nor volunteers from Berkeley High. You guys have phase separated over here, I can see. So what do you do? Well, typically what you would do is take your molecules into animal studies. Now, this, of course, is a very controversial practice. And especially in the city of Berkeley, we have a lot of very vocal opponents and also proponents of animal research. We have found in our laboratory that sometimes taking a non-classical approach to doing animal research is both beneficial to our psyches and our karma and also to our research. So in this particular circumstance, the first animal models that we tested our compounds in were not what you would consider the standard animal models for human cancer, which might be like laboratory mice or laboratory rats or whatever. Instead, we thought, well, maybe there's an organism that we can test these molecules in that will not be harmed by the molecules, and that also would lend itself towards the particular experiments that we want to do, which have to do with visualizing the sialic acids. We want to see these sugars. And what better organism to see sugars in than an organism that's transparent? So what organisms are transparent? The zebrafish. Now, zebrafish, they're tiny little fish. So it looks huge up here. Okay, this is larger than life. These things are actually half a centimeter. Okay, they're very small. But they are transparent, which makes them a wonderful organism to use for studying development of organs. Zebrafish are one of the most popular animal models for developmental biologists because you can just watch them from the point of conception through embryogenesis to the generation of a larva all the way to an adult fish. And we thought, well, why not use zebrafish as a model to test our imaging reagents? Zebrafish have sialic acid just like we do. They're vertebrates. They have all the bits and pieces that we have. They have hearts, lungs, brains, spleen. They have a spine, like many of us. And so much was known about their developmental program. Everyone knows exactly what to expect with zebrafish. And here's what they look like during early embryogenesis. And they're just, you can tell immediately if there's something wrong. So we thought this is a great organism to test our compounds, because we can find out if we can see the sialic acids, but we can also determine whether our compounds are toxic or harmful or just not tolerated well. So that's exactly what we did. We started studying our compounds in zebrafish. So in an early experiment, 2 graduate students, Scott Laughlin together with Jeremy Baskin, demonstrated that one can actually fertilize zebrafish oocytes and generate zebrafish embryos in the presence of our synthetic sugar that has an azide. And during embryogenesis, those embryos will take up the sugar, convert it to sialic acid, and display the azides on the cell surface, and then we can see them with our imaging reagents. So in this experiment, they fertilized the embryos and let them develop over the course of 4 days in the presence of the sugar. The azides were then resident in all of the sialic acids in the body of the embryo. And then they took those embryos and literally dipped them in a solution of dye-FO that was linked to a green fluorescent dye. And they just let that reaction proceed for a variety of time points. What you're looking at here in the top row, these are embryos, what we call control embryos, in which this azide sugar was left out of the media. So these are sort of normal embryos with normal sugars. The bottom are the embryos that actually incubated in the presence of this azide sugar. So these embryos have azides. And you can see, it's exactly what you think you're looking at. This is a profile of the head of a basically 4-day-old zebrafish larva. That's the eye. That's the nose. There's the mouth. And there's the gills over here. And even after just 1 minute of reaction with this reagent, you get a nice, bright fluorescent embryo, and the embryos only get brighter and more fluorescent with time. And in these control samples, you really don't see any fluorescence above a kind of background level. So this was very encouraging. But most importantly, we never saw any harmful effects of any of these reagents. These fish grew up and lived normal, healthy fish lives. They were fertile. They had babies of their own later on. And that was very encouraging. It also opened the door to do some really dramatic experiments where we could actually track how the sialic acids were moving around inside the embryos by labeling them with different colored dyes at different time points. So for example, Scott and Jeremy did a whole series of experiments where they took embryos and fertilized them and allowed them to develop in the presence of the azide sugar for several days. Then they would label those azides with a green fluorescent dye. But then those same embryos could be put back into the chamber with more azide sugar so that even more azides could be introduced. And those could then be labeled with a different color, like a red dye. And they could do this up to 3 times so that when they looked at these embryos, there would be 3 different colors representing 3 different time points during development. And we started this whole program in developmental biology surrounding this type of experiment. And I thought, this is a dramatic image. It's what we would call a dorsal view of the head of a 5-day-old embryo. Dorsal meaning it's sort of like the front, sort of looking underneath the chin, underneath the head. So here's the jaw, this sort of V-type structure. You can't really see the eyes. The eyes are up here. They're a little bit out of view, but you can probably see the nostrils. It's a little more dramatic without the lights on. I don't know if we have control over that. But in any event, you can see there's 3 colors here. There's some green, there's some blue, and there's some red. Those are all sialic acids that were formed at different time points during development. And from experiments like this, we could tell things like, oh, when are the nostrils formed? When are the gills formed? We could track all these things. It was pretty neat. In fact, you can even do 3-dimensional reconstructions of these fluorescent embryos. And hopefully you can see this. It's not quite as nice up here as it is on my computer, but this is kind of a surface representation of that same embryo head. Now you can really see the eyes. That's one of the eyes. That's the other eye. These are the 2 nostrils. Here's the jaw. But again, very dramatic pictures of embryogenesis through the window of sialic acid. So now we've got the chemistry. We figured out how to put the azides into the sialic acids. And of course, skip over that. The big question is, can we take this technology and translate it into the context of human clinical imaging? And remember that at the outset, our goal was to be able to detect the different kinds of sugars that are on tumors versus normal tissue. So what does that mean? Well, first of all, we need to take the concept from fluorescence imaging, which is what I've been showing you with zebrafish, now into the clinically relevant imaging modalities of MRI and PET. Because fluorescence, in all of its glory, doesn't work so well for organisms that are opaque. And that is certainly the case for human beings. So by contrast, MRI and PET are very good at penetrating into the middle of the human body effortlessly. And so we think our technology is general enough that we can apply it to these other modalities. But that is a direction that we are now taking. And then finally, zebrafish are a wonderful model organism, but eventually we do have to get this technology into the context of human beings. And how we get there is, that's one of the major challenges that academic labs face. Because, first of all, it's much more expensive to approach a human clinical trial than it is to do work with zebrafish in a laboratory. And secondly, you have to work in an infrastructure that has access to human patients and to physicians and to all the approval boards and so on that are required for that kind of translational research. So we're now in the process of basically trying to raise money and trying to find partners in more clinical settings and maybe biotechnology settings so that we can take what we've done in the laboratory and bring it closer to the people in this audience. So these are 2 challenges that we're taking on. And what I'm hoping is that they're people in the audience that might even want to get involved in this kind of research. I know there's some high school students here, maybe there's even some Cal undergrads, I don't know. But I think there's a very rich future in this for someone who's entrepreneurial enough to do chemistry in a human body. Okay. So with that, let me just thank the various people, some of whose work I mentioned tonight, but all of whom are very vital contributors to the research that we do. In the early days of this project, it was such pioneers as Jennifer Prescher, who was a graduate student in my lab, working side by side with Danielle Doobie, who was another grad student. And those were the two that developed a lot of these azide sugars and did some of the preliminary work in the chemical reactions that we use today. Both of them are now professors in their own right. Danielle is on the faculty at Bowdoin College, and Jen Prescher just joined the faculty at UC Irvine. And then they were followed by another set of talented graduate students, those being Scott Laughlin and Nick Agard. They both graduated within the last few years, and they're now postdoctoral fellows, actually both in the Bay Area. And then we had a very talented undergraduate, Jay Codelli, who did a lot of the chemistry of these DIFO compounds, and Jay is now a PhD student at Caltech. So these people have all left the lab, but there's certainly plenty of talented people that are still working away up there in Latimer Hall at the top of campus. Jeremy Baskin is the person who developed DIFO, and he continues to do a lot of work in Zebrafish, and he's been joined more recently by a younger student named Karen Dainert, and she's doing some spectacular work in developmental biology. What she's discovered is we can use these tools to follow changes in the sugars on embryonic stem cells as they start to mature and develop down particular lineages, as we call it. And then the cancer imaging challenge is right now in the hands of Pam Chang, and she is going to be finishing her thesis work within the next year. She'll be passing the torch to a younger student, Allison Cohen, who's working collaboratively with a postdoctoral fellow, Elena Goon, who did her PhD work at Stanford in their molecular imaging center. So hopefully these are names that will soon become very famous and to whom I will owe a great debt of gratitude. Then last but not least, we have a very talented group of undergraduates that are very active in this research, particularly Hitomi Nakamura, who's synthesizing compounds that we hope will be viable for human clinical studies. And I should finally mention that we work very closely with my colleague, Professor Sharon Amaker. She's in the molecular and cell biology department. She's an expert in zebrafish husbandry. So she knows how to grow embryos. And were it not for Sharon's input, none of us would be able to tell you which end was the head and which end was the tail. So we owe her a great debt of gratitude. So with that, let me wrap it up, and thank you very much for your attention this evening, and then open the floor to questions from the audience. Thank you. There's a waving hand in the back. Do you have a question? No? Okay. How about in the front?
I can't help wondering. There was an article, a front page article in the Chronicle a few weeks ago, perhaps. The sheer number of forms of sugar that are now on the market. Xylitol, sorbitol. It's just, I'm intimidated. I'm absolutely intimidated by it, and I have a lot of knowledge on sugars. I do know that xylitol is affirmatively good for the teeth, the third sugar, and of course cane sugar is affirmatively bad for the teeth, but I'm not sure I know a lot more than that. I mean, there's just so many forms of sugar, some of which I've come home with from Whole Foods without realizing that I've never heard of this packet of whatever. But do you have a roster of the various types of sugar now on the market that people should avoid or not avoid?
Okay, so I think the question is, what's up with all the sugars on the market? What are we supposed to think of all that? And which ones are good and which ones are bad, right? Something like that. And you mentioned a few of those sugar substitutes like xylitol, sorbitol, mannitol. Let me just tell you, anything that ends with tol, those are not actual sugars. They're sugar alcohols. They're made from sugars by a reduction process, but they're actually not like sugars per se. They're used in foods because they're sweet, but they don't contribute as many calories, although not zero, but they don't contribute as many calories as a bona fide sugar might contribute. So you find those substances in sugar-free, like gum, sugar-free ice cream, sugar-free cookies, and so on and so forth. Okay, so are they good for you or bad for you? It kind of depends on your intestinal makeup. Some of those sugar alcohols are not well tolerated by people. They can cause horrible intestinal distress. Anyone who's pigged out on a gallon of sugar-free ice cream has probably paid the price for the following 12 hours. Sounded like a good idea at the time, didn't it? But don't go there. So I would say a little bit of that stuff is certainly not going to harm you, but I just wouldn't make a diet out of it. I really think, when it comes to the foods that you eat, you should just keep it very simple and natural. If you can't find it in nature, then you should think twice about eating it. That's my instinct. But having said that, most of these sugar alcohols are not going to harm you. They can just cause you a lot of intestinal distress. Just eat sugar, but less of it. Yeah.
You concentrate on trying to use these complex sugars with sialic acid for imaging. Is anybody working on using that for the presence of these sugars for treatment? Like, for instance, trying to engineer viruses or something else that will attack those kind cells and attack them preferentially?
That's a great question. So the question is, as we are trying to exploit elevated sialic acid for imaging, for targeting imaging agents, could one target those cells using viruses or toxic substances, that would seek out the sialic acids and actually kill the cancer cells? That's a great question, and I think it's a very interesting idea, although it's a far more challenging problem than what we've been doing, which is the imaging challenge. The reason is that while these cancer cells may have elevated levels of sialic acid, all your normal healthy cells still have some sialic acid. So there's a differential that we are trying to detect. And it's much easier to try and detect differentials than to exploit differentials for toxic targeting or viral targeting. The reason is that the normal healthy cells have actually a lot of sialic acid. There can be millions to billions of copies of that sugar on a normal healthy cell. And so if the basal level is 1 billion, a tenfold increase, right, to 10 billion, the basal level is still so high that you would have to fine-tune your toxic substance or your virus to the point where, 1 billion was below the toxic threshold, but 10 billion was above the toxic threshold, and that's just very difficult. Since we're developing agents that are intrinsically non-toxic, we don't have to worry so much about the basal level being so high. So if we can conquer the imaging problem, then maybe we'd be bold enough to take on what we call the therapy targeting problem, but it's a much more difficult problem. So nobody's gotten there yet, but people have talked about it. Yeah?
When you were talking earlier about the sugar on the cell, and you were showing how the cells with the virus attaching to them, you can take Tamiflu and prevent it if you're in a high-risk group, right? And then you said it was very similar there with the cancer cells. Now, do you see maybe something like Tamiflu coming only for cancer, high-risk people, where you could take a preventative thing that would adhere to the cell and prevent?
That's a good question. So the question is, if you know that cancer cells have elevated levels of some sugar, could you design some kind of therapy that you could treat high-risk individuals with that might prevent that change in the sugars? Maybe, but the fact is, and this is one of the great sort of skeletons in the closet of this field, although we know that certain sugars are elevated in these cancers, we actually don't know why. And we don't know that that elevation actually has anything to do with the malignant behavior of the cancers. So in other words, seeing an elevation in sialic acid correlate with malignancy doesn't mean that the elevation in sialic acid causes malignancy. So maybe you could prevent the change in the sugars. That doesn't necessarily mean you'll prevent cancer from forming. We don't know. We actually don't know. So this is a big open area of investigation. Why did the sugars change? If you stop them from changing, do you stop cancer? Or is that just some ancillary consequence of malignancy that has nothing to do with the actual biology of the cancer cell? We don't know. So someone needs to work on that. There you go. There's a thesis project. Yeah.
So with diabetes,
Yeah, diabetes, so you know, that when I prefaced so the question is, what about the sugar coating in diabetes, right? And you're right. So first of all, there's different kinds of diabetes. And there's one form of diabetes in which the pancreas is destroyed, and as a result, we no longer can make insulin. And insulin is important because insulin allows us to take up and utilize glucose. Okay? So people who have diminished insulin generally have elevated blood glucose, and that can be toxic. So generally speaking, however, those people, the sugar coating on their cells is totally normal. And that's because glucose metabolism is sort of walled off and separated from the metabolism that builds the cell surface sugars. They're two fundamentally different metabolic machineries. In fact, glucose metabolism is generally walled off from all the other metabolism in your cell because it's so important. Your cell has figured out how to isolate it so that it doesn't interfere with other things, and vice versa. So those people with diabetes generally have, in all regards, their cell surface sugars are normal, but their glucose, which is in a separate category, is all over the place, and they can't regulate it because they've lost the ability to make insulin, at least in some types of diabetes. It's totally separate. Yeah?
Have you heard of the blood type diet, the book that was written in the 90s, and do you know anything about it?
Have I heard of the blood type diet book? Yes. What do I think about it? Yeah. Supposedly there was science behind it. Yeah. I mean, to be honest, I don't really know what to think about that. You know, I don't want to completely dismiss it because the truth is there's a lot we don't know about central metabolism. You know, and we discover new things all the time. And, you know, it's funny because metabolism research was all the rage in the 1950s, 1960s. And then it sort of fell out of favor during the molecular biology revolution. So in the 70s and 80s, biologists were all about DNA, splicing and dicing DNA and genetic engineering and engineering new organisms and deleting genes and adding genes and all that. And all those people that discovered all those like pathways you have to memorize in school. Remember citric acid cycle and all, remember anyone know what I'm talking about? Who took biochemistry? And you know what I'm saying? You had to memorize all these pathways. That stuff was really hot research, you know, in the 50s and 60s. Everyone was trying to figure out all the pathways. So interestingly, now, you know, fast forward a couple of decades, there's a renaissance of interest in metabolism because people are now much more aware of how complicated it is, how tightly linked it is to our genetics, you know, how many metabolic disorders we suffer from because we're eating strange diets. And so, you know, I think it would be sort of naive of me to dismiss a book that claims that you should alter your diet as a function of your blood type. I shouldn't dismiss it, but the truth is that it doesn't really jive with what, you know, I know about the link between genomics and metabolism. But there are other, you know, areas of metabolism that certainly are governed by our genomics. And we also know that there's a lot of what we call polymorphisms in the human gene pool. And so it's entirely possible that because of my particular genetic makeup, there are certain, you know, foods that I should have more of or less of compared to someone else. That's entirely possible. But I don't know that the blood type is the place to focus. Blood types are governed by a very small locus of genes. Oh, right here?
Yeah, I noticed in many of your trials with Zebrafish that you didn't actually do any of the tests with cancer cells themselves. And I'm wondering if you've actually done any trials where you use cancer cells in correlation with this new sugar that you come up with, and whether or not you actually saw a significant difference. Because I'm just thinking, you know, if you have 1 billion lights turned on, 10 billion, how big of a difference that's going to make?
That's a good question. So the question is, what have we done with human cancer cells beyond this zebrafish work? And in that one image I showed of the cultured human cancer cells, what I didn't show is if you culture other cells that are not cancerous, what we call primary human cells, you get a lot less of this fluorescent sialic acid. So if there's a 10-fold difference, you know, in two different cell types, you can generally see that. 10-fold is well within the discernible range of fluorescence microscopy. Even 2-fold, we would have been able to see. But again, seeing differences between cancer cells in a test tube is one thing, and actually seeing those differences inside the body of an organism is something very different. And for those kinds of experiments, we need more money and, and probably a partnership with an industrial entity. Like GE Healthcare, for example, is a company that's big into medical diagnostics. There are a number of biotech companies in South San Francisco that we've been in communication with, some of whom might be interested in sort of jointly sponsoring a next level type of experiment. But we do need to get to that level. We actually considered doing experiments where we put the cancers into the zebrafish and use the zebrafish as a host as a cancer model, so to speak. But really, I just don't think there's any point in doing that for two reasons. One, human cells just are not going to grow well inside a zebrafish. These two organisms are just too different. That's A. And then B, people have been curing cancer in mice for decades. Mice have nothing to worry about with respect to cancer. And I feel like, and likewise, people have been imaging cancers in mice for decades. And I feel like, I don't know that we have anything else to add to that. And so many of these reagents that look so great in mice just bomb when they get into human clinical settings. So my instinct with an imaging technology is that we should try and go directly into humans, because we're not dealing with a toxic drug here. So we don't have the same number of hurdles to cross with respect to toxicity, which would require a whole lot of animal studies. Rather, we're dealing with relatively non-toxic substances that are meant strictly for imaging, not for long-term therapy. These are one-shot deals. So the hurdle is lower for us, and so we don't necessarily have to go through all these potentially irrelevant animal models. I think we could go directly into humans. But we're not there. You're right. Yeah.
Is there anything that could try to get protection of the sugars that instead of being fluorescent would be visible by an MRI or PET?
Yes. So the question is there something we could attach to the sugars that instead of being fluorescent would be something you could see by MRI or by PET? And the answer is absolutely. And in fact, there are reagents that you can see by MRI that are already out there used in the clinic every day. And one of the most common is this compound called Magnavist. So Magnavist is a compound that has a little magnetic element in it. It's got an element called gadolinium. And it has 7 unpaired electrons, this gadolinium element. You know the periodic table? So if you look way down there at the bottom of the periodic table, that's the part everyone kind of forgets about. Gadolinium is down there. It's in the lanthanide series. So gadolinium 3+ has 7 unpaired electrons, which makes it a really strong little magnet. And if you put gadolinium into an organic molecule, you can then attach it to other things. So people use Magnavist all the time. If you have an MRI and they give you an IV line and they inject you with something, it's probably Magnavist. So we already did this. We took Magnavist and we hooked it to DIFO. So now we can target it to the sugars. So that's how we would do MRI. For PET, it's even easier. PET uses a radioactive element. It sees radioactivity. And the most common radioactive element in PET is fluorine of the 18 isotope, F18, as they call it. It's a positron emitter. And the reason that we put fluorines in DIFO is because we thought, oh, eventually we can make those radioactive fluorines. And then it intrinsically would be a PET imaging probe. So that's how we're going to do PET. So yes, these things are totally doable, and they're in the works. There's hands pointing right here. Yeah.
I actually have 2 questions. The first is, if cancer cells are relatively undifferentiated, and so are fetal cells, will fetal cells also have high levels of nucleic acid on the surface?
Yes. So the question is, if cancer cells are relatively undifferentiated, as they are famously known, do they have the same kinds of sugars as embryonic cells? And the answer is yes. That's a very astute observation that, in fact, that hypothesis came out about 6 or 7 years ago, and then people started looking, and lo and behold, some of these sugars that I showed on that previous slide, which previously had been thought of as cancer sugars, it turned out they were embryonic sugars. And they are normally present in embryonic tissues, but then they disappear. And when you're an adult, you don't see them anymore. But these cancers re-express them. Yep. These cells are de-differentiating with respect to their morphology, their genomics, their proteins, and their sugars. Yep.
Second question. In the drugs that are used to treat, to prevent flu, so to speak,
So the question then is, how do the anti-influenza drugs work? Those drugs that I showed are inhibitors of the enzyme that cuts the virus loose at the end of the life cycle. So in other words, the virus can still attach to the sugar, it can enter the cell, it can replicate inside the cell, and then it gets ready to set itself free, but it gets stuck. They bind in the active site of that enzyme, neuraminidase. They're active site competitive inhibitors. In fact, they're transition state analogs. That's how they were designed. And which is also why, in part, why the drugs have to be used before you get sick, because if you're already symptomatic, you know, the symptoms of the flu are not to do with the virus so much as your histamine response to the virus. So when you respond to the virus, your immune system makes you feel really sick. And if you've already gotten to that stage, you have so much virus in your body that, you know, blocking the exit from one cell to the next is just not enough to shut it down. You need your immune system at that point. But yeah, those drugs are transition state analogs. They bind to the active site of the enzyme. And in fact, they're also famous for another reason, because they were optimized using the crystal structure of the enzyme. And it's one of the first and few examples of what we call rational drug design, where you take the protein target, you get a structure of the protein using X-ray techniques, and you literally look in there and design the molecule to fit perfectly in there, and it works. Usually people do that and it doesn't work. But this time it worked. So it's a very famous example. Yeah.
I was getting a little worried about the lactose intolerance that you mentioned earlier. Since I eat a lot of dairy and they're kind of expensive, and I am lactose intolerant, I was wondering if all the dairy products that I ate is just going wasted down the drain.
Do you take lactase?
I don't.
No? You just suffer, basically. So it's true that if you are not taking lactase, and if you are lactose intolerant, when you eat lactose-laden foods, you are going to lose a lot of their sort of nutrition and also caloric value. That could be a good thing or a bad thing, depending on what you're doing. If you're picking out on an ice cream sundae, let it go, you know. But you're paying the price. Obviously, it's very uncomfortable. Yeah, you know. Also, the truth is that a lot of the foods that we think of as lactose-rich foods can be found in forms that are not so lactose-rich. So, for example, you know, milk, cow's milk has a lot of lactose, but goat's milk doesn't. And same with the cheeses. You know, cow's cheese has a lot of lactose, but goat cheese doesn't. So a lot of times, all you have to do is swap one source for another source, and you can drop the lactose by several-fold, and you'll be better off for it.
So the enzyme for the lactose, the body might not have the enzyme for the lactose, but they might have the enzyme for the protein, because the digestion process for the protein is different.
Totally different. Yeah, totally different. That's right. Yep. You're still getting the proteins, no problem. Yeah. So the first question is, you have a friend who has cancer, and your friend was told not to eat sugars because it would be bad for the progression of her disease? Well, oh, I see. So she was told that the sugar would cause the cancer to grow. What kind of cancer are we talking about? Abdominal. You know, again, I'm not a physician, I'm not an oncologist, so take my words with a grain of salt. My instinct is that it's very hard to perturb the fundamental growth mechanisms of a cell just from your diet. You know, you have so many mechanisms in your body that keep you at what we call homeostasis, which means that if you eat too much of something, your body's really good at getting rid of the excess, and it generally doesn't affect your fundamental functions. Also, proliferation of cancer cells, in my understanding, is that it's governed not so much by access to metabolic substrates or by energy. It's really governed by the intrinsic machinery of the cell becoming dysfunctional. So we have growth regulatory mechanisms that prevent our cells from proliferating under normal circumstances unless they're supposed to because of wound repair or something like that. Cancer cells have lost the ability to control their own growth, irrespective of how much sugar they have access to. So my instinct is that your friend does not need to worry about the sugar in her diet. In fact, if it makes her feel better, she should go ahead and eat that ice cream sundae. Okay? your second question had to do with how close are we to getting these compounds into a human clinical setting. And we have to do some toxicology testing before, we could actually apply for approval, like human subject approval. But, with the right industrial partner that could help us finance a human clinical study, we could be, I don't know, 2 years maybe, something like that. If things go well, maybe 2 years. Yeah.
You spoke about the need for money to make this go further. So talk about the funding you've had up to now, and also maybe some personal interest about how you got involved in this research when you were a high school or college student. What led you this way?
Okay. So the first question is, how have we funded our research up until this point? And mostly through grants from either federal agencies or private foundations that focus on cancer. So when I first joined the faculty, one of the very first grants that was funded in my lab was from the American Cancer Society. I had a Young Investigator Award, which is a 3-year award, which allowed me to hire my first 2 graduate students and a postdoctoral fellow. And then in the context of that grant, we were able to generate some preliminary data that then formed the substance of a larger grant application to the National Institutes of Health. So we have now, the state initiative to fund stem cell research. And we just recently were awarded a grant from the Department of Defense to study prostate cancer as a target for our imaging technology. So it's really been, money from a lot of different sources, private foundations, state foundations, government agencies, all together. And then the second question was, how did I get into research anyways, right? Starting from high school student? Is that what yeah. I had no clue in high school about science, or I didn't really have that kind of foresight. When I was in high school, my life revolved around music, mostly. I played in a band, and soccer. I was on the varsity soccer team. And so I got into Harvard because they recruited me to play soccer. That was great. And then when I got to college, I thought about majoring in music, and my parents said, no. They said, that's not a good idea. They told me to pick something more practical. And so I started out as a biology major, and then in my sophomore year, I took organic chemistry, which is a requirement of all biology majors, and I kind of fell in love with it. And so I switched my major to chemistry in my junior year in college. And then, but I was always interested in biology, so the sort of chemistry-biology combo was something that was kind of percolating in me in my college years, and then really solidified in graduate school. So that's what happened to me. But my first research experience was in college, really, and maybe it was the summer after my sophomore year. I worked in a lab over the summer. It was a disaster, actually. I had no idea what was going on. I worked for a professor at Harvard Medical School, and she matched me up with a postdoc who was supposed to help me, but he didn't speak any English. And then she disappeared to Cape Cod for the summer, and I was kind of wandering around on my own. So that was kind of a bust. But then in my senior year, I ended up working for a physical chemist, and that was a much better experience. And I got a lot of good mentorship, and so things got better. Anyways, if your first research experience is a disaster, don't give up, because it could get better. Yeah.
Individuals, the carriers, the system, everybody who's trying to make decisions. Do you see any effort going in that direction? I'm really wanting to be prudent here with this $1 trillion that's coming up the pipe.
Yeah.
How do we really exercise that funding to move things ahead?
Yeah, these are cosmic questions. So the question is. They're asking me to repeat all the questions so that they can record it on their YouTube video. So let me try and encapsulate that one, which is, we're hopefully going to have an economic stimulus package any second now. And when that happens, and it looks like it's going to, there'll be, allocations to federal agencies, and there'll be chunks of money that need to be spent on a very short time scale over the next 2 years, we think, and so, or shorter, And so everyone in these funding agencies is wondering, how can they get the most return for an investment that's a short-term investment? And as you pointed out, there are a lot of holes in our present healthcare system. There are holes with respect to access. There are holes with respect to technology. And there's a disconnect between the cost of certain practices and the ability of payers, who are ill, to have access to those tests. So, so if you have $1 trillion, should you invest it in a pie-in-the-sky technology that might not come to fruition for 20 years? Or today, should you make the current technologies available to as many people as possible? And the answer is yes. The answer is yes. And I think, and for a number of reasons. First of all, absolutely there should be more investment in making technologies that are already here today and powerful accessible to more people. And I say things like, oh, MRI, PET imaging, oh, these are routine clinical imaging techniques. That's true in San Francisco and Boston and LA and New York. It's not necessarily true. It's not even true in Bozeman, Montana. There are a lot of places in our own country where it's not true, and it's not at all true in developing nations. And so we live in this privileged high-tech environment here in the Bay Area where I can make new reagents and cross the bay and test them in all these great instruments. So right now, we should make an investment in portable PET imaging, right?
Best practices in investigating what works. Maybe the MRI isn't important if I come in and have a headache. And that isn't really well researched.
Okay, so basically,
More technology at it, that's going to solve the whole problem. We just have to say that very firmly.
Yeah.
Looking at what are the best practices.
So this is a good question to pose to, I think, someone who's in the business of healthcare delivery. Okay, but the truth is that my own take on this is that without making some investment in next generation technologies, we're basically shutting the window, to opportunities. And I say this coming from a family that's riddled with cancer. My father had colon cancer and then bladder cancer, and my mother has had breast cancer twice. My father's sister died of colon cancer, and her cousin has colon cancer. His uncle had colon cancer, and my grandmother had breast cancer. So my days are numbered. I've already had 3 colonoscopies. I'm only 42. Okay, so I'm incentivized. My take on this is that as a chemist, I have certain insights that might allow me to develop a better diagnostic tool. Okay. I don't have insights into how to make current technologies better disseminated, better chosen. I don't have insights into those problems. I don't feel like I have anything to contribute there beyond and above what the smart people around me are already contributing. So, we each have a niche. And I feel like where my talents are best served is in taking chemistry into biology and trying to come up with an idea that is just hanging from a tree and no one's plucked it. So that's my niche.
Time for one more question.
Oh, I get that. Azide is a common preservative in a lot of lab reagents, so I'm concerned about the toxicity that when you incorporate it into humans. So what are people's thoughts on that?
Yeah, this is a common question that I get. And so the question is, isn't azide a preservative, and it's a toxin, and it blocks, it's a metabolic poison, and why would you inject that into a normal, healthy human being? That's true for azide anion, N3-. So N3- is, like you said, a metabolic poison. But alkyl azide, so when you put N3 on an organic molecule, it's inert. It's like the difference between, so like one analogy I could use is like hydroxide. OH- is a strong base, but if it's CH3CH2OH, it's ethanol. We drink it, right? So it's the same kind of thing. So N3- is fundamentally different from like an alkyl azide. And in fact, there's many drugs on the market today that have alkyl azides, and the azide is just inert, just kind of like goes along for the ride, like AZT. You know, AZT has an azide in it, and it's not, that part of it isn't intrinsically toxic. It goes through your body unprocessed. And so these azides of ours are really very non-toxic. We shot them into plenty of animals, not humans, just animals, and we've never had any kind of poisonous response. They're really just sugars. Okay.